Thanks for the introduction. So as mentioned, I'm going to talk about network alignment, which in a sense is another way to perform a cross-species protein function prediction. So I'll get into these concepts during the presentation. So suppose we have some human proteins. Here I'm just listing 8 for simplicity. And of course, for some of them, we know what their functions are. So that's what the colors represent. But for many others, we do not know their functions. So that's what the gray circles are. As we all know, however, we want to study what these proteins do to better understand human disease, human aging, etc. But performing experiments is often difficult, much more difficult than doing them in model species. So one way to get around this is we can actually transfer knowledge from said model species, such as yeast, to human. And one computational technique to do so is sequence alignment. So given a bunch of yeast proteins, a bunch of human proteins, we compare all their sequences. And if we find those that are sequence similar enough, we may consider them to be orthologs and hypothesize that they have similar function. So for example, if we compared all of these proteins together, let's suppose that we found B2, C3, E5, F6, G7 to be sequence similar. And then, for example, we could infer that protein 2 has the green function. However, in our studies, we've actually found that around 40% of orthologs, human yeast orthologs from the YeastMind database, do not actually share function with respect to the current gene ontology functional data. So obviously, this is an issue if we want to use sequence alignment to transfer knowledge. Similarly, it's possible that many sequence dissimilar proteins can still share function. For example, A and 1, we didn't find them to be sequence similar, but they still could share function, or maybe they could not. So we need more. And one way to get this additional information is through networks. So as we all know, proteins interact with each other, and these interactions are ultimately what carry out function. So for example, these proteins may come together. We may model them as a yeast PPI network, where we have this green unit up here performing some function, and this mostly yellow unit down here performing some other function. And similarly for human. Then, if we want to compare PPI networks, then we need network alignment, where analogous to sequence alignment, but now we have our 2 PPI networks. We don't know which proteins correspond to each other. Our goal is to try to align not only similar proteins to each other, but also proteins that have similar neighbors, and similar neighbors' neighbors, and so on to each other. And so the idea is that we're trying to preserve the interactions that occur. And this idea of topological similarity can be used in tandem with sequence similarity to generate our alignment. And so now we can use this alignment to transfer knowledge between the species. For example, we might predict that protein 1 and 2 have the green function, 7 and 8 have the yellow function, and then also go in the other direction, we may predict that F also has the yellow function. However, network alignment still has its issues as well. In particular, oftentimes the aligned nodes do not correspond to proteins that perform the same function. So in all of my previous examples, of course, they were the same color, they did perform the same function. I just used that to sort of illustrate the basic ideas of network alignment. But in actuality, what may happen, for example, taking a part of our previous network, is that D and 4, E and 5, these proteins may not actually share the same function, even though they're aligned to each other. And so again, this brings up the question, can we use network alignment to transfer knowledge if this happens? And so, of course, in order to address this issue, we want to understand why aligned nodes are not sharing the same function. And one possible reason is that many current network alignment methods assume that topologically similar nodes have high functional relatedness. So I briefly alluded to topological similarity above, but now I'll explain it further. Basically, the idea is that nodes with similar neighborhood structure are similar themselves. And so in these two highlighted nodes, they are topologically similar because they are both a part of many triangles. On the other hand, these two nodes are less similar because, maybe while they have the same degree, this one's a part of triangles, whereas this one's a part of squares. So their greater neighborhood structure is not as similar. And then these nodes would be even less similar because now the degrees are not even matching, and the nodes don't even loop back on themselves, for example. So again, current network alignment methods assume topologically similar nodes have high functional relatedness. We want to investigate if this assumption actually holds or not. We do so in two tests. So first, we compare a network to an identical copy of itself. So in this situation, we know that the most topologically similar nodes, in other words, AA, BB, CC, etc., are those that are functionally related, because of course they're the same node. So we want to see basically if we align the most topologically similar nodes, will they be functionally related? So we look at the topological similarity between every pair of nodes across these two networks, and we visualize them with the figure on the right. So basically, the x-axis is our topological similarity score, and the y-axis is the percent of pairs with a given similarity. As we can see, all the functionally related pairs have the maximum possible similarity. Again, expected because the two networks are identical. And the functionally unrelated pairs, their similarities are spread out all across. Basically, this is telling us if we align the most topologically similar nodes, we will be aligning the functionally related ones. And so this is sort of saying that our analysis confirms what we expect. Now, the more interesting part is if we compare a network to a 20% dissimilar version, which basically means that they're 80% similar in the sense that they're sharing 4 out of their 5 edges. And now, again, we perform similar analysis as before, looking at the similarities, and we find that the functionally related pairs are actually only slightly more similar to each other than they are to the functionally unrelated pairs. Basically saying, if we were to align the most topologically similar pairs, there's not actually a very high chance that they would be functionally related. So basically, this assumption does not hold very well. Thus, a paradigm shift is needed with how the network alignment problem itself is approached. So we propose a data-driven framework called TARA, which essentially learns from the data what should be aligned, using this idea of topological relatedness, which I will discuss. So again, we have our yeast PPI network. We have this green unit performing some function, this yellow unit performing some other function. Now, through the evolutionary process from our common ancestor, maybe a gene duplication event occurs. In human, this green unit may actually have an extra protein in it for whatever reason. And similarly, this yellow unit may lose an interaction because it wasn't so important. So if we use traditional network alignment methods to align these two networks, what will actually happen is that this yellow part will be aligned to this green part because they are both a square with a diagonal down the middle. In essence, the traditional network alignment methods are trying to solve this, are mathematically motivated. They're trying to solve this subgraph isomorphism problem where they're trying to directly match the topologies of the two networks. On the other hand, we sort of have a biologically motivated approach where we know that these green proteins perform the same function, so we're trying to learn that, for example, a triangle in yeast becomes a square with a diagonal in human, a square with a diagonal in yeast becomes a square in a human. And then if we were to see a green triangle in another part of the yeast PPI network, we would try to align it to a square with a diagonal in another part of the human PPI network. And so this idea of learning from the data, predicting what's functionally related and not, naturally lends itself to a classification framework, which I've outlined here. So I won't go into too many details about these. For that, you can come to my poster, but I will briefly talk about each of these steps. So essentially, we start with our two networks, yeast and human PPI networks. And then we need to capture the topological and possibly sequence patterns between the yeast and human proteins. So like the example above, we want to capture that, for example, one protein is part of a triangle and the other protein is a part of a square with a diagonal. And then, of course, given two proteins, we need to determine if they're functionally related or not. And we use the gene ontology information to do this, where basically, if two proteins share at least k Go terms, we consider them functionally related. Otherwise, they're not. And then, with our features and our labels, we plug this into a classification-based network alignment framework, where essentially, as with classification, we split our data into training and testing sets. So to perform the alignment, we look at a testing set, and anything predicted to be functionally related, we just take as part of the alignment. And this is to avoid any circular argument. And now, actually, as a side effect of this, because we are not including any part of the training set in our alignment, this actually leads to our method not being able to theoretically attain a precision of 1, just because we're missing out on potentially predicting on some of the data. But as we'll see shortly, this doesn't actually impact our method too much, and we're still able to outperform the existing methods. So and then with our alignment, we use an established protein function prediction framework for network alignment, essentially. So here's a brief overview of our results. We compare to 3 other state-of-the-art network alignment methods, Wave, Sana, and Primaline. And to explain what's going on here, basically, by default, Primaline uses both topology and sequence information. So naturally, we wanted to create a topology-only version to make it more fairly comparable to the other methods, including ours. And we also added the method called sequence, was essentially we took the sequence information from Primaline and treated that as the alignment to see how well that would do. And as we can see, our method Tara performs the best. And it even outperforms the Primaline version that uses both topology and sequence, which is very promising for us because we're working on incorporating sequence information into Tara, and that could just potentially be even better results. Now, another interesting side effect of our study is we found that when we use Primaline with only topology, we see a precision of around 0.05. And when we use the sequence information from Primaline, we also see a precision of around 0.05. And yet, somehow, when used together in Primaline, the precision is actually much higher than simply the sum. So it's at 0.3 instead of 0.1, which is suggesting that if we can somehow effectively incorporate sequence information, for example, into our method, we could even see even better results than one might expect. So essentially, in the study, we've challenged the current paradigm of network alignment, which assumes that topologically similar nodes should be aligned. Instead, we say, learn what should be aligned. Our method does exactly that and is able to outperform the existing network alignment methods in the task of protein function prediction. As such, we've provided a valuable data-driven framework for these tasks. And lastly, I would like to briefly mention there are two network alignment methods that also incorporate functional knowledge, like us, called Tana and Monk. References are down here. Tana, as of now, has not been published yet, so we've not been able to compare against it. And we have done some preliminary comparisons against Monk, and we have shown and we've seen that our method does better, but of course we need to be more thorough. But I guess the main point is that while this direction of data-driven network alignment is still in its infancy, many people are starting to pick up on it, and we encourage anyone who's studying network alignment to pursue this direction. And then, of course, my poster is M45, which will happen tonight. If you have any more detailed questions about the methodology and all that, feel free to come and ask me questions. Thank you.
Thank you very much. Are there any questions from the audience? Yes, please.
Could you give a few details about the test that you did? So what were the types of proteins? How many proteins did you look? How were they annotated?
Can you, like, which test do you mean?
The last slide where you showed your scores.
Like this?
Yeah.
Right. So we have human PPI network, yeast PPI network, and we use the different methods to align them. And then based on this alignment, we make predictions about...
The question is, what are the networks? Which types of proteins do you look at?
What do you
Is
mean?
this synthetic data? Or?
No, so these are actual like physical PPI interactions between the
so signaling networks or metabolics or PPI
protein interaction networks. Yeah.
Okay.
Any other questions from the audience? Yes, please.
Following up on that other question, so a lot of PPI networks are inferred from orthology, so it'll break, I suppose, your original assumption of not using alignment. But do you think you could try this method with other PPI networks that are built from orthology?
Yeah, I mean, this framework is general in the sense that we can sort of plug in any types of networks, and as long as we have some functional information, we can make predictions, I guess. So, yeah.
Yes, please.
You started with the notion that is there are only 40% of the functions shared between yeast and human. But after this method is applied, what kind of improvement do you get? And also, is it through all three aspects of gene ontology, or you just focus on one of them, like molecular function, or?
Right. So in these, we focus on biological process. So that branch of gene ontology.
But the problem is yeast is a single cell, while human is multicellular, and there are a lot of processes in the human that didn't exist in yeast.
Right.
So that you won't be able to predict.
Right.
Yes, please.
Can you give us a little bit of information about the complexity of your method?
Right. So I guess there are a couple parts. So one is actually getting the features of the node pair. So to do that, we actually count graphlets. I don't know if you're familiar. That's basically like counting the small subgraphs, like motifs and such. So that is fairly complex. But then, of course, the classification part is quite quick, just because in general we don't find that many pairs are functionally related. So our training test sets are quite small usually.
Any more questions from the audience? Then maybe I have a question. So you mentioned that adding the sequence information might actually further improve your approach. Do you already have an idea of how you would include it in your approach now? And are you planning to do it or try it?
We are planning to do it. We haven't really explored too much into that direction yet, so can't really give you an answer to that. But yeah.
Great, thank you. We have maybe one more question, very shortly.
It's a very quick question. Can your method handle weighted graphs where it's not just a binary edge, it's there or not, but you have weights, confidence scores, things like that, which are present in many networks?
Right. So in theory, our method could, just because we could swap out this topological feature and instead, use a weighted feature or something. So in theory, yes, we could, but we haven't specifically studied weighted networks.
Great. Thank you very much.